Hi everyone, welcome to my channel CAPSA. So today's topic is about ELISA, which is a very important topic. So it is a laboratory diagnostic procedure for detection of various toxins like diphtheria toxins. And ELISA can also detect specific antibodies like IgG, IgM, IgA, etc. And it is considered that the most sensitive test for the diagnosis of HIV and various serological testing. Now, what is the time needed for this test? So the time actually takes 2 to 3 hours, but for rapid test takes 10 to 20 minutes. Enzyme-linked means enzyme is used to label one of the component of immunoassay. It can be antigen or antibody. And immunosorbent means an absorbing material used that specifically absorbs the antigen or antibody present in the serum. And the absorbing material can be polystyrene or polyphenol. Now, what are the types? So, 4 types: direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. In case of direct ELISA, this technique actually used for the detection of antigen. And the enzyme labeled with primary antibody in case of direct ELISA. In case of indirect ELISA, this technique actually used for the detection of antigen or it can be antibody. And enzyme labeled with secondary antibody. In case of sandwich ELISA, this technique used for the detection of antigen mainly. And the enzyme labeled with primary antibody in case of sandwich direct ELISA, and enzyme labeled with secondary antibody in case of sandwich indirect ELISA. So competitive ELISA used for the detection of antigen or antibody, and enzyme labeled with secondary antibody. Now, what is primary antibody and what is secondary antibody? So primary antibody actually directed against the antigen, but secondary antibody is an anti-human immunoglobulin which can directed against Fc region of any human or other species of immunoglobulin. Okay. Now, what are the principle and procedure? So the principle, it actually based on the immunochemical principle of antigen-antibody reaction. Now, what are the procedure or what are the steps involved in this process? So in case of first step, an inert solid surface is used, that is polystyrene, made up of polystyrene or polyphenol. So antibody against the protein or antigen to be determined is fixed on an inert solid surface which is made up of polystyrene. In case of second step, what is done? The biological sample or patient sample containing the protein or the unidentified antigen to be estimated is applied on the antibody coated surface. So these proteins can be antigen which is unidentified antigen. Okay. In case of third step, what is done? Now this protein antibody complex is then reacted with second protein specific antibody or antigen specific antibody to which an enzyme is covalently linked. Okay. This enzyme must be easily assayable and gives a product which is colored product. Now, after adding the substrate, it gives the color change of the product. Obviously, after washing the unbound antibody linked enzyme, this substrate will be added. Okay. After adding the substrate, the color change of the product will be occurred, and this color change actually detected by using spectrophotometer. So this color change is detected by spectrophotometer. Every time after washing, unbound part will be removed. Okay. So this is the ELISA technique. I hope you enjoyed the lecture. If you like my video, Please do like, comment, share and subscribe. Thank you for your attention.